Hi everyone, this is your instructor Paramecium, and this is part one of our lecture on the classification of eukaryotes. This lecture would cover mainly classification of protozoans and a brief touch on algae classification as well. By way of review, it's important that we remember the important features of eukaryotic cells generally in terms of the plasma membrane, the organelles, and cell division, mitosis, and meiosis. So a review of the general characteristics of protozoans, fungi, algae, and animals is important to understand and follow this lecture. So protozoans are a very diverse group made up of three characteristics. They are eukaryotic, meaning they have membrane-bound organelles. But in addition to that, they are unicellular and they lack a cell wall. So unlike plants and algae that have cell walls, they do not have cell walls. Additionally, they may require specific environments like fluid, such as ponds, or an environment such as the soil. And we have few of them that are pathogenic, that is, they can cause diseases. Morphologically, they have the characteristics of the eukaryotic cell. They show variations in terms of their nucleus, because you have some that have more than one nucleus. You have some that have differences in size of their nucleus. Then there is a mitochondrial variation, vacuole presence, motility as well. And so we'll use some of these features to group or classify the protozoans. Generally, protozoans are motile. They have a motile feeding stage known as the trophozoite. It is also the growth stage. Some of them have a resting stage where they form a cyst. Most of them are chemoheterotrophic, or what we will call saprophytic. So they rely on organic molecules as a source of both their energy and their carbon. But you have very few of them that are photoautotropic. So those that have the agar characteristics, they're able to create their own food, or synthesize their own food using sunlight as a source of their energy, and carbon as a source, inorganic carbon from carbon dioxide as a source of their carbon. Reproductively, they can reproduce as either sexually or asexually. Asexual reproduction may occur through schizogony. For schizogony, the main difference between schizogony and binary fission is that for schizogony, before the cell starts dividing, the nucleus divides into several different or into a number of nuclei. So you have all these nuclei, and these are known as scissions. So based on the number of nuclei that exist, the cell will divide into an equal number as well, so that you have kinesis, which is the division of the nucleus, and then it will follow by the division of the cytoplasm. There are 7 novel or new ways of classifying protozoans based on their rRNA sequences. Remember, the rRNA sequences are some of the most conserved regions of organisms, so it's relied on heavily. Previously, there were 4 old kingdoms that were used to classify protozoans. So we have Sarcodina, Mastigophora, Celiophora, and Sporozoa. But the new taxa groups the protozoans by Parabasal Ovulates, and the ovulates will cover the ciliates, the epicomplexans, and the Dinoflagellates. Secosa Radiolaria, Amiibosua, Euglinosua, and Diplomonadida. There are three basic life cycles that protozoans exhibit. So they may be haplontic, diplontic, or diplohaplontic. Now I want you to pay attention to the first part of these definitions. So for haplontic, it's referring to the haploid number of chromosomes in the vegetative stage. So the definitions refer to the number of chromosomes in the vegetative stage. So haplontic will refer to vegetative cells that are haploid, and the zygote being the only diploid cell in the life cycle. And then for diplontic, the vegetative cells are diploid, the gametes being the only haploid cells in the life cycle. And then diplohaplontic would alternate between haplontic and diplontic. And so if you have an organism that has, if we represent the diplontic by 2n, for haplontics, the vegetative cells would be n, and the zygote would be 2n. Okay. And it's the reverse for diplontic, where the vegetative cells is 2n and the gametes would be n. So if we look at each of them separately, the groups that I just mentioned, Parabasala, they lack mitochondria. They lack mitochondria. They have what we call a parabasal body, or Golgi bodies. And representatives of this group are Trichonympha. Trichonympha is a symbiotic flagellate that is found in many species of termites. So they actually help the termites in its survival. And then you have Trichomonas or Trichomonas, which causes trichomoniasis. An example would be Trichomonas vaginalis, which also is a causative organism for trichomoniasis. And this is how trichomoniasis will present in the vagina. Then you have the ovulates. The ovulates are tubular. They have an ovuli, and these are protists or protozoans that are characterized by the presence of sac-like, a fluid in a sac-like membrane. And these sacs, which are enclosed by lipid or fats, are then called alveoli, and that's how the name comes by. There are several or a number of subdivisions. You have the ciliates, the epicomplexans, and the dinoflagellates. So this is a presentation of an alveoli, alveolus in terms of singular. So if you look at the ciliates, they are chemoheterotrophs. So they will rely on chemicals as a source of their energy, and they rely on organic molecules as a source of their carbon. The ciliates are binucleate. So the ciliates will have two nuclei. So that's a very important characteristic feature. Representatives will be Verticella, Balantidium, and Paramecium. For Verticella, this is how it presents. It's like it appears like a vortex. So this is how anatomically it presents. It's able to suck in food in the environment through the currents that is in the stream that it finds itself in. And then you have Balantidium coli. Balantidium coli is an example of an organism which belongs to this group. Then you have Paramecium. Paramecium exhibits what we call conjugation. Remember, we've already come across conjugation, but this conjugation is different from that in bacteria. In this conjugation, you have divisions of the micronuclei. So Paramecium has both a macronucleus, a bigger one, and a micronucleus. And during conjugation, you have two organisms, a pair, coming together, and there are divisions of the micronucleus, and then it is then transferred. There's an exchange of the micronucleus. Then those micronucleus will then fuse, the macronucleus will disintegrate, and then one of the micronucleus will then become a macronucleus, and then the cycle continues. So there is a very distinct difference between this conjugation and the one that occurs in bacteria. Then we also have the epicomplexans. The epicomplexans they have what we call an epicoplast, which help them to attach to their hosts and to penetrate their hosts. And so they are parasitic in nature. They may form spores as part of their life cycle. Important examples are those that belong to the genus Plasmodium, Cryptosporidium, and Toxoplasma. So I have presentations of the three. So you have Plasmodium, and Plasmodium would occur intracellularly in the cells, like red blood cells, like rings. So you have this here, that's how it presents. Cryptosporidium on the surface of the gut epithelium, that's how it presents. And then you have Toxoplasma, which is more extracellular in terms of its presentation. Then the last group under the Ovulates are the Dinoflagellates. They are photosynthetic in nature, but their names come from the fact that they have two flagella. So we call them bioflagellates or dinoflagellates. A large proportion of them are plankton, so plants-like in nature. They may produce some bioluminescence. They may also produce some pigments, and some of them will also produce neurotoxins. So dinoflagellates appear this way. They have two flagella. There is one here, and there is one that goes across its body. And the pigments that they present, this is red pigments that's covering the surface of a water body that was produced by dinoflagellates. Then you have Secosa. So we've already looked at Parabasal, we've looked at Ovulates, and now we're looking at Secosa. Secosa are amoebae. That is, they have pseudopodia. They are amoeba-like in nature. They have these false legs, and so they are amorphous. So there's a very distinctive characteristic that their body changes shape. Their shells are made up of calcium carbonate, and representatives of the group will be Foraminifera. You see the different shapes that it assumes in all these from A to Q. You see all the different shapes and the pseudopodia that is being used differently in these organisms. And then you have Radiolaria. So Radiolaria are also amoebae in nature. They also have pseudopodia. But the distinct feature that they do have is that they are radially centered. And their shells are made up of silicates. They are radially centered in that the way they present, if you should cut through them, it gives you a radial presentation, a mirror image. It's like an orange. So you have an orange. This is where you have the attachment to the stalk. If you should cut it through like this is radially centered. So that's how they also present. Then you have Amiibos. Amiibosua are slime molds. They are plasmodial. That is, they don't have they are acellular, so you can't really say they have cells. In terms of their life cycle, they have body parts that are not compartmentalized. They have a diploid nuclei, but they practice what we call streaming cytoplasm. So their cytoplasm is able to stream its content, move it around, so that there is a fair distribution of food and oxygen to all parts of the cell. Very fascinating. You see some form of it in plants, which we call cytoplasmic streaming. The plasmodia becomes plasmodial masses, and then they will divide into sporangia or single cells. They will form stalks. They undergo meiosis in their spore forms or sporangia. And then these spores will then develop into what we call myxoamoebae and then fuse together to form the plasmodia again. So I have the cycle here I'm going to show you briefly. And so this is how they appear, the slimes. Over a number of hours, up to 24 hours, you would see an entire cycle unfolding. So this is the spores that have fused myxoamoebae, and then it would then undergo mitosis. There is aggregation, and then it starts migrates, becomes a slug, and then there is a culmination, and then eventually you have the spores being formed, the sporangia becoming haploid spores, and then it will fuse again and the cycle will continue. Then under protozoans, another group that we have in terms of taxa is the Euglinosua. So they have modified flagella. The most important characteristic is that they have disc-shaped cristae in their mitochondria. So they are mitochondria, but the cristae that are the surface of the inner walls of the mitochondria are shaped like a disc. There are two types. We have the Euglinotes and the Kinetoplastics. The Euglinotes were for a long time identified as plants, misidentified as plants for a long time. They are photoeutrophic, so they have chlorophyll. They can synthesize their own food using sunlight through the process of photosynthesis. But when the sunlight is not available, they would practice chemoautotrophism. So what they would do is that they would rather use chemicals as a source of their energy, but then will still be able to synthesize their food. They use paramylon. So note this, they use paramylon instead of starch as their storage form of carbohydrates. Morphologically, they don't have a cell wall, but they have flagella. It helps them in moving around. They also have a red eye spot for directional movements. So you see this red eye spot here. It helps them in moving in certain directions. Interiorly, they have structures called pellicle that fortifies their cell membrane. They're able to reproduce mitotically and by cytokinesis. And then they form cysts under harsh conditions, which is also a very important point. So an example would be Euglena gracilis. Then the kinetoplastics, they have a single mitochondria. So they have one mitochondria, and the mitochondria is specialized. So there is specialized kinetoplastic at the ends of the mitochondria. They are parasitic, pathogenic as well. So some of the diseases that they cause would be trypanosomiasis or sleepy sickness, and then leishmaniasis. The second picture here shows leishmaniasis, and these are the organisms, the parasites in blood. So these are extracellular parasites. Okay. Then you have the Diplomonadida. They lack mitochondria, they lack ribosomes, they lack peroxisomes, but they also have two nuclei, so we call them binucleates, and they may have several flagella, so they are multiflagellated. And a good example is Giardia lamblia. So this is Giardia lamblia. You see the many flagella that is emanating from the body of the organism. So for our participation question for this week, Giardia lamblia and Trichomonas vaginalis. So Giardia lamblia is already spelled here. Trichomonas. Trichomonas vaginalis. Trichomonas vaginalis are examples of medically important protozoans that do not have mitochondria. How do organisms like these obtain energy without mitochondria? So again, Giardia lamblia and Trichomonas vaginalis are examples of medically important protozoans that do not have mitochondria. How do organisms like these obtain energy without mitochondria? So you may have to read around a little bit, and based on your previous understanding of metabolism, give a good response to this question. The last slide is on algae. So algae are mostly phototrophics, and you find most of them in the oceans. They are unicellular, and they store energy in the form of oil. So if you look at the diatoms, the group of algae, they are unicellular. They will store energy in the form of oil, and they have an important component, the domoic acid, which may cause neurological diseases. So ingestion of mussels that have fed on diatoms may affect other organisms like birds and sea lions and any other organism in the water that feeds on them. Then you have the dinoflagellates, the planktons. Some may also produce neurotoxins like the red tide, as you see in the picture. And these, from time to time, may kill fish, marine organisms, and even humans may suffer from parasitic shellfish poisoning if we consume food that is contaminated with a toxin. So this brings us to the end of part 1 of our lecture on the classification of eukaryotes.